everyone, this is Miss Moriarty, and I'm here to discuss with us an instructional video on DNA and its structure, as well as the scientists who discovered DNA's structure. Remember, as you watch this video, you want to make sure that you take down notes using the follow-along notes that I posted to Classroom, or writing it down on a separate piece of paper with a pen or a pencil. Also, note that because we are asynchronous for today, you want to make sure that you are watching this video on Edpuzzle, answering the questions, and completing it for attendance. So here we go. Right, so we know that DNA is the molecule of life. All living things are going to contain DNA, right? Remember, that is a characteristic of life. It's going to be found in the nucleus of all eukaryotic cells, and we know it contains the genetic code that's going to determine our physical features or traits that get passed down through generations. Now, DNA actually stands for a really long name, and it's called deoxyribonucleic acid, but it actually can tell us two things from that very long name. One being that DNA contains a 5-carbon sugar called deoxyribose, and it's also found in the nucleic acid category of our macromolecules, which means it is made up of what's known as nucleotides, right? Remember, a nucleotide is a monomer or a building block of a nucleic acid. You can kind of see that in our DNA molecule here, right? The DNA as a whole is a nucleic acid, while the parts that make it up are known as its nucleotides. Now, nucleotides consist of 3 parts, and this should look really familiar to us because recall, ATP, or adenosine triphosphate, is also a nucleic acid, and it's a nucleotide. And so we see that nucleotides consist of 3 things: that sugar bonded to a phosphate, bonded to a then nitrogen base. Now, in DNA, there are 4 possible nitrogen bases that we can find, and it's those 4 different bases that allow for genetic diversity. And if you think back to our recipe for traits dog activity, you can remember within the DNA strips, there were only 4 symbols, but it was those 4 symbols that produced all different types of dogs in our class. And it's the 4 nitrogen bases known as adenine, guanine, cytosine, and thymine that help to make that genetic diversity. But it's those base pairing rules that help us pair up those nitrogen bases when we see them in the DNA molecule. Because remember, it's double-stranded. So there's 2 strands of those nitrogen bases that are going to be connected. Now, it's adenine that always pairs up with thymine and vice versa. And guanine always pairs up with cytosine. And a really good way to remember this is, if you think about it, apples we find in trees, right? A, apples, T, trees, adenine A goes with thymine T. We can also always see cars, cytosine C, are always found in the garage, guanine G. It's just a good way to remember it. Now, the reason why they pair up the way they do is because of their molecular structures. And those 4 bases actually fall into 2 different categories. The first category is known as a purine, meaning that this structure has a double ring. And it's our adenine and guanines that are classified as purines, while our thymine and cytosine are classified as pyrimidines, meaning they only have single ring structures. And so a purine always is going to bond with a pyrimidine as per their base pairing rule. Another way to remember which one is which, you always think opposites, right? The bigger the word, the smaller the molecule. Another good hint, or another way to remember what is what, if you notice, the word pyrimidine has a Y in it. So does our thymine, so does our cytosine. But remember, the reason why a thymine and a cytosine is a pyrimidine is not because there's a Y in their name, it's because they are single ring structures. But remembering that there's a Y in their name is just a good way to remember their pyrimidines. Now, who discovered this? There was actually a scientist by the name of Erwin Chargaff. He actually found out and stated that DNA from any cell should have a 1-to-1 ratio between its pyrimidines and purines. So that's where we found out we had a base pairing rule. And his discoveries actually helped Watson and Crick develop their model of what's known as the double helix. And so actually using his rule, if we are given a certain number of cytosine, guanine, thymine, and adenine, we can determine how many are at each. So here, if our DNA molecule contains 28% cytosine, we can find out then how much guanine, thymine, and adenine are present. So we know that there's 28% cytosine. So how much guanine will there be? Well, we know that cytosine always bonds with guanine, and there's a 1-to-1 ratio, so there's most likely going to be about 28% guanine. But how much of the DNA have we accounted so far, right? 28 plus 28 is going to give us about 56% of the molecule. And so this tells us that 44% of the molecule must be made up of adenine and thymine. And if you can recall their base pairing rule, we know adenine and thymine go together. And so if we took that 44% and divided it by 2, that should give us 22% for each. Now, I mentioned before that it was Erwin Chargaff who discovered our base pairing rule and that assisted both Watson and Crick in discovering DNA structure. Because we actually didn't always know what the structure of DNA was. And it was in 1953 where we saw James Watson and Francis Crick determine its shape using an X-ray diffraction picture. And though Watson and Crick tend to get most of the credit for discovering the DNA, it was also the work of some other scientists, specifically Rosalind Franklin, who took that picture or took that X-ray diffraction picture of DNA that allowed them to determine that DNA has a double helix structure. However, because Rosalind Franklin at the time was a woman, there were some issues that happened between a lot of the scientists involved. And it was also Maurice Wilkins who also contributed, but didn't necessarily get along too well with Rosalind Franklin. And so all of those findings now tells us that DNA is a two-stranded helical molecule. And when we unwind it, sometimes it looks like a staircase, sometimes it looks like a twisted ladder. But there's a reason why when you unwind it, it will look like a ladder, and we'll get into that soon. So when we unwind and we know our then molecule looks similar to a ladder, we can actually break down the certain parts here. So the sides of the ladder, here are our sides, are comprised of our deoxyribose sugar and our phosphates. And actually we call that the sugar phosphate backbone. And it's the nitrogen bases that are going to make up the rungs of the ladder. So it's our A, T's, C's, and G's that are going to make up the rungs or the steps of our ladder. And it's going to be hydrogen bonds that's going to hold our strands and hold our nitrogen bases together. And if you notice here in this picture, our adenine and thymine are going to have 2 hydrogen bonds holding each other together, while guanine and cytosine actually have 3. And so it's this phenomena here that tells us DNA has actually the same width throughout the molecule. But our adenine and thymines and guanine and cytosines all have different widths, but the fact that they pair up with opposite structures of each other is what allows DNA to then have the same width throughout. And the reason why our hydrogen bonds hold our DNA together is because eventually when our cells need to make more, they need to actually unzip the DNA in order to replicate it. And so the hydrogen bonds that hold our DNA together are actually quite weak. And so then that brings us to DNA replication, which we will talk about a little bit later on in the week. All right, guys, make sure that you answered the questions in this video, you wrote down notes, and please mark this as done because remember, it's a part of your attendance.